Now that we've learnt about the cell cycle and about mitosis, the next section we're going into is how do cells actually control that whole cycle. So our learning intentions are that the cell cycle is controlled using three main checkpoints, which we'll learn today. And then if the cell cycle goes wrong, that can lead to abnormal cell behavior, in particular having cancer. So after this video, you should be able to describe the three main checkpoints that cells use to regulate the cycle in eukaryotes, and then explain how mutations in two different types of genes lead to cancer. First of all, if we couldn't control the cell cycle, it would just go on and on and on, and cells would keep dividing forever, and that would cause a whole lot of problems for our bodies. So tumours growing for cancer is basically uncontrolled cell growth, and other diseases like this one here, which is called psoriasis, is caused by malfunctions in the cell cycle. You can see psoriasis, it looks a little bit like eczema, and it's just layers and layers of skin cells growing on top of each other because the cell cycle's gone out of control. So here are the checkpoints in the cell cycle. There's three main checkpoints that cells use, and they're basically like a stop sign like we've got here, and the cell needs to make sure everything's okay before it moves on to the next stage. So the main jobs of the checkpoints is to check that the DNA, which is the genome, or the information in the DNA, is to make sure that it's complete and there's no damage in the DNA before it gets passed on to its two new daughter cells in mitosis. If any errors get detected, then the cell cycle can stop at one of our three checkpoints and hopefully be repaired. So our three main checkpoints are the G1, which happens near the end of G1, the G2 checkpoint near the end of G2, and the mitosis checkpoint, which happens at metaphase. So let's learn the details for all of those. In the G1 checkpoint, remember, once we finish G1, we're about to go into S phase and copy our chromosomes. So the most important thing at this checkpoint is to make sure that we're ready to replicate our DNA. So the cell checks for any damage in the DNA, and it checks that there aren't any breaks or any mutations. If everything's okay and it's favorable conditions, then we go ahead and we go and do our S phase and we copy our chromosomes. If not, we stop there and hopefully repair the damage. But if it can't be repaired, sometimes the cell goes off into a side phase called G0, where the cell rests and it doesn't divide, and it keeps attempting to try and repair it. It might end up dying through apoptosis, where the cell basically destroys itself, and we'll learn that in the next section, next lesson. So we've gone past the G1 checkpoint. We've made a second copy of our DNA. So all our chromosomes have sister chromatids. We're going through the G2 phase. So the cell's continuing to grow. It's continuing to just do its job, being whatever cell it is, like it's being a skin cell and making all the proteins that it needs. So before we get into mitosis, you need to get past the G2 checkpoint. So the most important thing it does here is to check the copied DNA is ready to go and there's no mistakes in the copying. So it checks that DNA, checks for any damage, and if we're okay, we can go ahead and go on and start mitosis and hopefully divide into two perfectly nice copies. While mitosis is happening though, because it's such a complicated process, there's another checkpoint that's evolved, and it happens in the metaphase. So remember, in the metaphase, our chromosomes have lined up down the metaphase plate, nicely lined up, with our spindle fibers from the centrioles attached to each one at the centromere. So at this checkpoint, there are some proteins that are checking that the sister chromatids are attached to the correct spindle fiber, ready to be pulled apart. And then once that check is done, and we go ahead and pass the checkpoint, then we go into anaphase, and those sister chromatids get pulled apart into opposite directions by those spindle fibers. So that is our 3 main checkpoints. The next section is going into a bit more detail about how those checkpoints actually get controlled in the cell. So don't forget that every process in cells, or basically every process, is controlled by proteins. Proteins are the worker molecules. They do pretty much everything in the cell. They can speed up chemical reactions, they can send messages, they can check for DNA damage, they can carry things. Proteins do everything. So the proteins involved here, they're in 2 categories, and I like to think of the analogy of like a car. So we've got go and stop. So let's talk about how this all works together and what these proteins do. So the first category are called proto-oncogenes. Onco is to do with cancer. So oncogenes are cancer-causing genes, but normally these aren't cancer-causing. So proto means they're not cancer-causing genes yet. So when they're doing the right thing, these genes are useful because they make proteins, and these proteins help the cell cycle to progress forward when it should be, as long as the right conditions are met. So proto-oncogenes are like the accelerator pedal in the car. They make the cell cycle go forward. Here's our accelerator. There's another category of genes called cancer suppression genes. And as the name suggests, their job is to stop cancer from happening. So the proteins that these genes make are there to stop the cell cycle at each checkpoint and make sure everything happens and everything's repaired before we go on. So the cancer suppression genes, if we're thinking like a car, they're more like the brake pedal that's going to stop the cell cycle at the right place. So we've got our accelerator genes and we've got our brake genes. The problem is that some of these genes can be mutated just like any gene. So mutations in either of these 2 types of genes are going to lead to cancer. And let's use the car analogy to figure it out. So with the proto-oncogenes, if they have a mutation, then they can't do their job. So they're like having the accelerator stuck forward, and the cell cycle is just going to keep on going and keep on going. Obviously in a car, sticking the accelerator forward, you're going to go and have a crash eventually. You don't want to have the cell cycle going on forever. And if a cancer suppression gene has a mutation in it, well then it's not going to be able to stop the cell cycle where it needs to be. So if you're in your car and your brake pedal isn't working, you can't stop when you need to, and again, you're going to go off and have a crash. So you need to be able to start the cell cycle at the right time, and you need to be able to stop it at the right time. So if these 2 categories of genes aren't working, then cancer is the end result. Going back to proto-oncogenes quickly, I didn't mention if a proto-oncogene is mutated, then it becomes an oncogene. So that's where the name comes from. And that's how scientists actually discovered them. When they go and look at the genes in cancers, in people's tumours, they actually find a whole lot of these genes with mutations in that shouldn't be there. And so we discover them first by seeing that they cause cancer when they're mutated. All right, let's look at a little bit more detail about those horrible diseases that are cancer. So like we've talked about, since the cell cycle's out of control, then cells are going to keep dividing uncontrollably, and that's how a tumour forms. So we've got here a typical smoker's lung. You can see all the black tar that's built up in this person's lungs from so much smoking. And this big white section here is a tumour in their lung cancer, which is their lung cancer. Doctors and scientists have come up with 4 different stages for how progressive a tumour is. The first 3 stages, well, we start at stage 0, and obviously the larger the tumour, the greater the stage. And once someone's cancer gets to stage 4, that's actually called metastatic, and that's where individual tumour cells are actually able to break off and go through the bloodstream, and they attach to other organs, and then they start to grow even more tumours. And that's the really horrible stage of the illness, where people are in pain and they have no energy. And often stage 4 cancer is incurable once it gets to being metastatic. The other interesting thing and dangerous thing about cancer cells is how they don't do this process called contact inhibition. So here's some normal non-cancerous cells that scientists put on a petri dish, and they've just grown them out on the petri dish, and the cells, because they're normal, once they bump into each other, they stop dividing once they've made a nice neat layer. So this is what the layer looks like if you zoom in. But cancer cells, and that layer, once the cells bump into each other and stop growing, that's called contact inhibition. But cancer cells don't show contact inhibition. So if they get grown on a plate like this, or in the body as well, they just keep on dividing and keep on dividing, and they make just layers and layers of cells, and that's how tumours form. It's random and they're not controlled, and it's just a whole lot of mass of cells that shouldn't be there at all. Alright, let's look at an example in a bit of detail about one of these cancer suppression genes. So remember, the suppression genes are the brake pedal. So here's one of our brake pedal genes, and it's called P53. So the job of P53 is it is here to monitor that G2 checkpoint. So remember, at the G2 checkpoint, the whole point is we're checking that our chromosome has copied into 2 perfect copies for our sister chromatids. And the G2 protein, it comes along and it actually attaches to the DNA in the chromosome. And you can see here's the protein, and in the middle you can actually see the double helix of the DNA, which P53 is attached to, running through the middle here. So the protein comes along and it attaches to the DNA, and that stops cell division, and then some other proteins come along and make sure that the DNA gets repaired in the right place. And so if the protein gets mutated, it can't attach to the DNA anymore, and then it can't do its job, and the cell cycle just goes ahead even if there's some mistakes. And scientists have found that in about 50% of cancers, this P53 gene has a mutation in it, and it can't do its normal job to stop at the G2 checkpoint. And that's pretty typical of a whole lot of these cancer suppression genes. Okay, the last thing we'll talk about is mutations, because I've mentioned them a little bit, and that's what's causing our 2 types of genes to malfunction. So what actually are mutations? Well, mutations are a change in the DNA sequence. Remember, DNA is a molecule and it's got 4 bases or 4 letters in its genetic code, which are G, A, T, and C. And it's that genetic code, the order of the bases, that make up our genes, and that's how our cells get controlled. So a mutation is just a change in one of those bases in the code. You can see here we had a T in the original sequence, and some sort of mutation has caused there to be a C now. And that's enough, often, to cause a gene to malfunction completely, and then the protein can't work, and something bad happens to the cell. Mutagens is the last keyword, and these are just factors in the environment that cause a mutation. So every time we're exposed to these things, there's a chance our DNA gets damaged and we're causing mutations in our cells. So there's radiation, of course UV light from sunlight, that's why we need to wear our high SPF sunscreen, because otherwise we're going to mutate our skin cells and get skin cancer. X-rays are dangerous. Lots of carcinogens like cigarettes. Even processed foods have a high chance of mutating DNA. Lots of strong chemicals like cleaning products, and some viruses and bacteria can also cause mutations in our genes. Alright, so that is controlling the cell cycle, and you should be able to explain the two types of genes and what they do to control the cell cycle.